Next section, poxvirus, specifically monkeypox virus. So the previous slide here, actually no, the next slide here and this one come from the same article. The link to the article is right down there, so you can copy that from the PowerPoint if you want to go have a look. It's a nice one on monkeypox, sort of a good general review in one of the Nature journals. So what are some of the other things that monkeypox is doing? Well, so this is meant to be monkeypox virus. It's going to block activation of the complement cascade, which if you've had immunology, you know that's kind of a basic level set of equipment that's running around in your bloodstream. They detect and destroy all kinds of bad things, bacteria and viruses, generally before you know they're there. In this case, virus shuts it down. It can't be activated, or at least attempts to shut it down. There's always a trade-off. IRF3 signaling and NF-kappa B signaling. Those are things that are downstream of RIG-I and MDA5, but they're also downstream of other processes. So, yeah, that's a thing. These pattern recognition receptors are things like toll-like receptors. Again, if you've had immunology, they can detect parts of exploded cells is a good way to put it. So like things that should not be in your bloodstream ever. They're there and they will detect it, or they'll sometimes be inside of a cell that grabs things and eats them for a living. So we can just check out, like, what did I eat? And, if it binds to one of these, then you say, oh no, something is wrong, and they go out and try and solve the problem. Virus will also block interferon alpha and beta signaling, because of course it will. A lot of viruses invest a lot of their genetic material in preventing this, because interferons are generally good antiviral molecules. At least when the body's making them. We'll escape apoptosis in those cells, so you can squeeze a couple more baby virus copies out before the cell dies. Fantastic. Okay. What's the virus going to do? Well, it can come in a number of different ways. So the virus can come in through the respiratory tract, and so here it is coming in there, or it can come in via the skin. If you get little pustules on there, if you have some kind of break in the skin in particular, then that can definitely happen. Once the virus gets in, if it's able to come into contact with a dendritic cell or a macrophage, it will infect those. And these are both, again, part of the immune system. And they're sort of the very abundant and kind of early acting parts of the immune system, particularly the dendritic cells. They reach out and do a lot to kind of regulate how the rest of the immune system is going to respond to a particular threat. Anyway, these cells know what to do. So when there is danger, when they pick up danger signals, they will go and they will run to basically a draining lymph node. And of course, in this case, they're already carrying the virus, so they have become essentially Trojan horses. Yeah, isn't that terrible? So they're going to go in there, and in the lymph node is where you would find tons of other white blood cells of various types. And so that then can allow the infection to spread, and you can end up with all kinds of bad stuff. Here we're showing the virus getting in through some kind of wound, totally another way that it can happen. We know that virus can come out in an aerosol from some people. Some will have ulcers on their tongue from the virus, and then as you're breathing out, you are breathing out virus. Yeah, monkeypox. And we know that some people have monkeypox virus in their semen. That's also true for several other viruses. There's even a reasonable evidence in a couple of cases that people were infected from seminally transmitted Ebola virus. Yeah. So far, I am not aware of any direct evidence for this transmission of monkeypox virus, but if it's in there, it could happen. It could totally happen. So, yeah. Great. Another wonderful thing about the world, right? The respiratory consequences of monkeypox virus are very bad very quickly. Now, we don't have much information on what happens in people, but it is believed to be relatively similar. This is what happens in monkeys. So these are, I believe, macaque monkeys. And they infected 15 of them through the respiratory route, and all 15 of them died very quickly. It did not take many active copies of the virus at all to be a lethal dose in monkeys, which is worrying. They developed lesions on the inside of their mouths, so they could probably spread it to each other, even if they weren't all there in there together. And they developed all these other things. So some problems on the skin. This is the rash. But all of them also developed bronchopneumonia. So you get a horrendous immune reaction in the lungs in particular. And problems down in the gastrointestinal tract, just problems basically all over the body. So monkeypox virus and poxviruses in general are considered to be very promiscuous viruses in that they will infect a lot of different hosts and a lot of different tissues inside of a host. Most other viruses are more restricted. They're only able to get into certain tissues and only able to work in certain hosts, but pox viruses are a big risk for being able to jump species to species. Essentially, there are cases where they don't have to change at all in order to do that. All right, what's in a pox virus? Well, we looked at the genes before. That was at the genetic level. Here are some of the proteins that the virus makes and roughly what they do and where they go. So this is what the virus looks like, the virion rather. It's got a little cage in here, and the cage is going to be packaging the viral DNA, and the viral DNA is all wrapped up with lots of proteins. And then on the outside, there are as many as 3 different membranes on this virus. And so one membrane is going to be trapped in here under this layer of protein D13. Another one of these is going to be out here, and another one's out here. The virus has an array of proteins, and there are often different proteins on each of these membranes. And the thing is, I believe all these membrane-coated forms of the virus are able to infect, but potentially they're able to infect different cells, which is not great. Yeah, not great news at all. And so some collection of these appear to be spike-like or envelope-like proteins in that they can bind to a receptor and somehow induce membrane fusion of the virus into the host. Now, on the mature virion, which is this kind that has 2 outer envelopes, if I remember correctly, this one has this entire group. So there's 9 proteins that make a complex, we think. If you take out any of those 9 proteins, I believe the other ones no longer work. But it looks as though these are 9 ways, potentially, for the virus to grab onto a cell, because each of them looks like it may have something like a receptor binding domain. There are other components over here in this little dotted window that appear to, in some cases, associate with those proteins or assist in entry and fusion. So that's crazy. Instead of having just, one spike like a coronavirus, these things will have 9, 10, 11, maybe 12 things that do that job. And then on the outer membrane of the envelope virus, you actually have several more proteins that stick out and are potentially involved in receptor binding, or sticking to some aspect of the host cell in such a way that it's going to lead to a successful receptor binding event. Yeah. So down here we can see what the one of those, the D13, is doing. The virus actually starts out as an icosahedral virus, and it turns into that thing. Here's some pictures over here of the immature virus. This will be inside of cells, and then it'll sort of collapse and dehydrate a bit. And you'll end up with this core, so that's going to be where the viral DNA is. You'll have kind of a pretty thick wall that goes around the core, and then you'll have these lateral bodies that are packed with different kinds of proteins, and then you'll have your outer membranes, different numbers of outer membranes. So the core is made of D13. It is icosahedral all by itself. So it makes a little, basically 20-sided ball, um, like a polio virion, but bigger. Yeah. And then that will associate with a membrane from, usually from the Golgi apparatus, and it will wrap that membrane around itself. And so this is the wrapping process, also carried out by D13 in addition to some other things. And so in this way, it's going to pick up two membranes. And now if it goes out of the cell without breaking the cell membrane, it can actually pick up a third outer membrane on there. Yeah, which is pretty crazy. Have variable numbers of membranes like that. But this is how it works. And these are from a technique called deep etch electron microscopy, which produces, I think, the coolest images in science. Look at that. You can see how crowded and just chaotic the inside of the cell is. And you can see all the little parts here that are basically, caught and frozen and fixed, but in the act of assembling themselves. And there you can see it almost completed, and there it's completed, and there's a different completed one sitting next to a mitochondrion. Look at that. It's got the little stripes on there, like a little baby tiger. Yeah, fantastic. All right, so that's a good place to end this section. And then we're going to go into a little more about, we'll eventually get to what we can do about the virus. And I think that'll be a good section to get to.